Cyanobacteria are one of the unsung heroes of life on Earth. They first evolved to perform photosynthesis about 2.4 billion years ago, pumping tons of oxygen into the atmosphere. A period known as the Great Oxygenation Event, which enabled the evolution of multicellular life forms. Led by BTI faculty member Fay-Wei Li, researchers have discovered a new species of Cyanobacteria, Anthocerotibacter panamensis, which could help illuminate how photosynthesis evolved to create the world as we know it. The work was published in Current Biology on May 13. "We never intended to discover a new species," said Li. It was a total accident. His lab was working on a project to isolate cyanobacteria from hornwort plants, and noticed something weird in a sample from a rainforest in Panama. The researchers sequenced the cyanobacterium's DNA, and found that it belonged to a group called Gloeobacteria, which is extremely rare. Prior to this discovery, only 2 species of Gloeobacteria had been isolated, Li said. There is also a third group of uncultured species from the Arctic and Antarctic regions, but no one knows how many species are in that group. Gloeobacteria diverged from the more commonly studied Phycobacteria about 2 billion years ago. The two groups have many differences, and a panamensis shares some traits with each. Similar to other Gloeobacteria, the new species lacks thylakoids, the membrane-bound compartments that are the site of the light-dependent reactions of photosynthesis in Phycobacteria and plants. Now we can be pretty sure that the thylakoid evolved in Phycobacteria, Li said. On the other hand, a panamensis makes carotenoids, a group of compounds that help protect an organism from sun damage, in a fashion similar to Phycobacteria and plants, but different from the other Gloeobacteria. These results suggest that this particular carotenoid biosynthesis pathway evolved in the ancestor of all cyanobacteria, and then was lost in some Gloeobacteria, said Li. Li said one of the more interesting findings is that a panamensis has very few genes that encode the proteins that perform light-dependent reactions. The researchers found that the new species could still perform photosynthesis, but very slowly, which could be of interest to synthetic biologists. If you want to build a complete set of photosynthetic machinery with the fewest necessary components, then this species could inform how to do that, said Li. Anthocerotibacter has a minimal set of photosystem subunits, but it still functions. Story source. Materials provided by Boyce Thompson Institute. Original written by Aaron J. Bucci. Note: content may be edited for style and length. Thanks for watching. Please subscribe my channel for more updates.